We are going to discuss about bacterial morphology. Okay, we will first discuss bacterial cell wall, then we will move for bacterial membrane and many more. So first, starting with the bacterial cell wall, because cell wall of bacteria is very important. Okay, so we will discuss cell wall of bacteria into two groups. First, we will discuss cell wall of gram positive bacteria. Okay, cell wall of gram positive bacteria is simpler in compared to gram negative bacteria, and gram positive bacteria contain peptidoglycan layer, very important. Okay, cell wall consists of peptidoglycan. So first we will discuss about peptidoglycan. So in gram positive bacteria, the peptidoglycan layer is much thicker, about 60 to 80 nanometer. Clear? Now each layer, and this peptidoglycan layer, number of layers is about 50 to 100. About 50 to 100 layers of peptidoglycan is found. So each layer is a mucopeptide, that is murin chain, which is composed of alternate units of NAM and NAG, that is N-acetyl muramic acid, N-acetyl glucosamine. So each layer is mucopeptide composed of alternate units of NAM and NAG. Clear? And they are cross-linked to each other via two things. They are cross-linked to each other via two things. The first one is your tetrapeptide side chain, and pentaglycine bridges. Okay, so these alternate units are also cross-linked with the help of tetrapeptide chain and pentaglycine bridges. Okay, now coming to the tetrapeptide chain. So tetrapeptide side chain is composed of, first one is your L-alanine, then D-glutamine, then L-lysine, and then D-alanine. Okay, first one is your L-alanine, then D-alanine, and you can remember like this. Okay, AGLA, in Hindi agla. So L-alanine, G for glutamine, L for lysine, and A for alanine again, but of D configuration. Okay, this is the composition of tetrapeptide chain if you ascend from any NAM molecule. Now coming to the pentaglycine bridges. So pentaglycine bridges just like this. Okay, 5 glycine will be there. Clear? Now the L-lysine of one tetrapeptide chain is covalently linked to the terminal D-alanine of the adjacent chain via pentaglycine bridges. Okay, suppose this is your tetrapeptide chain. Okay, suppose this is your L-alanine, D-glutamine, L-lysine, and D-alanine. Clear? And here another same D-glutamine, okay, L-lysine and D-alanine. Simple. So the L-lysine of one, this L-lysine of one, okay, this L-lysine is connected by pentaglycine bridges, which we have discussed here. Okay, with the D-alanine here as like this. So pentaglycine is connecting these two tetrapeptide chain, and these two tetrapeptide chain are linked with NAM. Okay, this is also linked with NAM. So this is the basic structure of the peptidoglycan. Clear? So one second revision, tetrapeptide chain consists of L-alanine, alanine, glutamine, lysine, and alanine, and there is a pentaglycine bridge. Okay, now moving to the second component of the gram positive cell wall is your teichoic acid. This is a second component of gram positive cell wall. So gram positive cell wall contains significant amount of teichoic acid, which is absent in gram negative bacteria. It is absent in gram negative bacteria, and teichoic acid is polymer of glycerol. It is a polymer of glycerol or ribitol, which is joined by phosphate group. Okay, so teichoic acid are the polymers of glycerol or ribitol, which are joined together by phosphate group. Clear? Okay, now the functions of these molecules are not still clear, but they may be important in maintaining the structure of cell wall. That is all. Now teichoic acid are again of two types. Teichoic acid are divided into two types. The first one is your cell wall teichoic acid. Okay, and the another one is your lipoteichoic acid. So the cell wall teichoic acid is covalently linked to NAM molecule of peptidoglycan. If any teichoic acid is directly linked with NAM molecule of peptidoglycan, that will be known as cell wall teichoic acid. And if it is attached to lipid group of cell membrane means if teichoic acid is attached with cell membrane, it will be known as lipoteichic acid. lipo teichoic acid. If it will cell wall, then cell wall teichoic acid. Simple. Okay. Now we will discuss about gram negative bacteria cell wall. Okay. So gram negative cell wall is first thinner but more complex. It is thinner but more complex than gram positive cell wall. Okay, and it comprises many components. The first component is your same, that is your peptidoglycan layer. Okay, but here the peptidoglycan layer is thin, that is 1 to 2 layer. Number of layers are 1 to 2. Here number of layers were 50 to 100. Number of layer is, so peptidoglycan layer number of layer is 1 to 2 and 2 nanometer thick only. Clear? Now, and coming to the structure of peptidoglycan, so it is same. It is made up of NAG and NAM molecules. Okay, but there are 1 or 2 differences which I need to mention in peptidoglycan layer. The first difference is that presence of meso diamino pymalic acid. There is presence of meso diamino pymalic acid. Very important. So meso diamino pymalic acid is present at third position of tetrapeptide side chain. Here third position was occupied by lysine. Third position by occupied by lysine, but when you will talk about gram negative bacteria, then its third position is occupied by meso diamino pymalic acid. What is the name? Meso diamino pymalic acid. Clear? No problem here. Now the next important difference, that in gram negative bacteria there is no pentaglycine bridge. Means pentaglycine bridge is absent in gram negative bacteria. So first difference is meso diamino pymalic acid, then pentaglycine bridge is also absent. Clear? And here the tetrapeptide side chain are directly linked to each other by the covalent linkage between D-alanine of one chain with meso diamino pymalic acid of the adjacent chain. Okay. So coming to the discussion of membrane. So there are 2 membrane present in gram negative. Okay, one interior to the cell wall and one exterior to the cell wall. Whereas in case of gram positive, only interior membrane is present. So first coming to the outer membrane in case of gram negative bacteria. So this is a phospholipid layer which lies outside the thin peptidoglycan layer, and it is firmly attached to the latter, means firmly attached to the peptidoglycan layer by a special membrane protein that is known as bronze lipoprotein. So this is very important. Okay, I have told you that suppose this is your cell wall of gram negative bacteria, peptidoglycan layer, one membrane is present here and one membrane is present here, and this is outer membrane, this is inner membrane, and then outer membrane and this peptidoglycan layer is connected by membrane protein that is known as bronze lipoprotein. Okay, so this is very important point, bronze lipoprotein. Clear? Now what are the functions of outer membrane? So outer membrane is act as protective barrier. So this outer membrane is providing protective barrier to the cell. Also, coming to the next point, that is porine proteins. So they are the specialized protein which is present in outer membrane, and 3 porine molecules cluster together and span the outer membrane to form a narrow channel. Okay, narrow channel through which your molecules which is smaller than 600 to 710 daltons can pass. Okay, like this 3 units will mix together and it will form a narrow chain through which molecule will pass. Okay, the next important point, so large molecules like vitamin B must be transported across the outer membrane by a specific carrier. Clear? So these are the functions of the outer membrane. Then coming to next important point, that is LPS, lipopolysaccharide. So this layer is very unique to gram negative bacteria. Okay, this layer, lipopolysaccharide layer, is unique for gram negative bacteria which is absent in gram positive bacteria. Okay, now it consists of 3 parts. This lipopolysaccharide consists of 3 parts. So the first one is your known as lipid A, or it is also called as endotoxin. Okay, the second part is core polysaccharide. The second part is your core polysaccharide, and the third part is known as O side chain and also known as O antigen. Clear? Now we will discuss each of them. So first coming to the lipid A, it is known as endotoxin. So it has endotoxic activities such as pyrogenicity, lethal effect, tissue necrosis, anti-complementarity activity. Clear? Now this is the activity of this. Now how it is composed, what are the composition of lipid A, we will discuss that. So lipid A is consists of 2 glucosamine sugar. It consists of 2 glucosamine sugar derivatives. Okay, each with 3 fatty acid and phosphate attached. 2 glucosamine sugar and each of them with 3 fatty acid and phosphate attached. Clear? Second important point about lipid A, that it is buried in the outer membrane. Okay, this part, lipid A part, will be buried in outer membrane, and the remaining part, this remaining part will project outward from the outer membrane. Clear? Now coming to the core cell core polysaccharide. So core polysaccharide, it is projected from lipid A region and it is composed of 10 to 12 sugar moieties. Okay, coming to the O side chain, this is very important. So O side chain is also known as O antigen. Clear? It is a polysaccharide chain extending outwards from the core polysaccharide region, and it is made up of several sugar moieties, and it greatly varies in composition between different bacterial strain. Okay, and O antigen is major surface antigen. It is a major surface antigen which is found in bacteria, also called as somatic antigen. Okay, and if this is antigen, then it can induce antibody formation. Okay, so it can be used in for stereotyping. So this is the use of O side chain. Clear? So this is about your lipopolysaccharide. Now moving to the next part. Next part is your periplasmic space. So no more thing, just you remember that it is the space between the inner cell membrane and outer membrane, which can, okay, I have told you here that this is your outer membrane, peptidoglycan layer, and inner membrane. So inner membrane and outer membrane, the space, okay, this is space is known as periplasmic space, here, which consists of peptidoglycan layer. Now how you will demonstrate cell wall? Coming to that, so the cell wall cannot be seen by light microscope and also does not stain with simple dyes. Very important, simple dyes, you cannot use simple dyes, and it can also not be seen by light microscope. So how we will demonstrate? So the first method you can use for this is plasmolysis. Okay, when bacteria is placed in hypertonic saline, shrinkage of the cytoplasm occurs while cell wall retains original shape inside. The next is you can do differential staining. You can look for electron microscopy. So these are the methods through which you can look for cell wall. Clear? So this is all about your cell wall and outer membrane. Now next part we will discuss is your cell membrane. Cell membrane, all of you are knowing from your earlier studies. Okay, so I will not go in detail of the cell membrane. So cell membrane, I am going for the important points, and the most accepted model to describe cell membrane is your fluid mosaic model. Clear? Okay, and it is 5 to 10 nanometer thick, composed of bilayer of phospholipid, in which several proteins are also embedded, which are known as integral proteins or peripheral proteins. Clear? Now one important point which I need to mention here, which is a basic difference from eukaryotic membrane. Okay, eukaryotic and prokaryotic membrane is somewhat similar, but one or two differences are there. So the one difference is that lacking of sterols. Prokaryotic organism such as bacteria lacks sterol in their cell membrane. Okay, instead of sterol, it has pentacyclic sterol like molecule. What is pentacyclic sterol like molecules that are known as hopenoids? So this is important point. Hopenoids are present in bacteria. Clear? And some carbohydrates are often attached to in the outer surface of the plasma membrane proteins. This is not so important. Okay, coming to the functions of cell membrane. So it is a semi-permeable membrane. It is responsible for acting as transport system. It is the site for metabolic process also. Okay, so this is all about your cell membrane and cell wall. Now coming to the cytoplasmic matrix. So bacterial cytoplasm, unlike that of eukaryotes, lack membrane bound cell organelle. You are knowing. Okay, the cytoplasmic matrix is mainly composed of water. About 70% of bacterial mass is water, and is packed with water. It packed with ribosomes. It has storage granules which are called as inclusions, and cell membrane invagination, which is called as mesosomes. So you will find these materials in your cytoplasm. Okay, in bacterial cytoplasm. It means they lack true cytoskeleton. It also lack true cytoskeleton. Okay, but have a cytoskeleton like system of proteins, but true cytoskeleton is absent. Now the one more term which I need to mention here, that is what is protoplast. So protoplast is just plasma membrane, and everything within it is known as protoplast. Clear? Now we will individually discuss all the components. So first coming to ribosomes. Okay, so ribosomes are the sites for protein synthesis. These are composed of rRNA and ribosomal protein. So ribosomes are made up of rRNA plus proteins, and this is the site for protein synthesis. Ribosomes are integrated with, okay, one more important point, this ribosome when integrated with mRNA and lead to formation of polysomes. Okay, many ribosomes will integrate with mRNA and lead to form polysome, name poly, it's a indicate name. The next important point, that they are 10 to 20 nanometer in size with a sedimentation constant of 70S. It means ribosome is of 70S, which is present in bacteria. you are knowing in prokaryote 70S ribosome is found, and 70S has two components, two subunit, that is 30S and 50S. Clear? Now after ribosome we will discuss for next, that is inclusions. Okay, intracytoplasmic inclusion. So they are the storage site of nutrient. First you can define what is inclusion body, why they are need. So it is a storage site of nutrient and energy which is present in some bacteria. Okay, and they are formed by bacteria under nutritional deficiency. When there will be nutritional deficiency, and then inclusion bodies are formed, and it disappear when deficiency is over. Okay, when deficient nutrients are supplied, then this inclusion body is over. Clear? Now there are two types of inclusion bodies. The first one is your organic inclusion body, and the second one is your inorganic inclusion bodies. So these are the two types of inclusion body: organic inclusion bodies and inorganic inclusion bodies. Clear? Coming to the organic inclusion body, so it includes your glycogen granules and polyhydroxy butyrate granules. Clear? So organic inclusion bodies include glycogen granules and polyhydroxy butyrate granules, while inorganic inclusion body, it includes polymethaphosphate or volutin or metachromatic granules. Okay, this type of granules will be found in inorganic inclusion body. Clear? The next and one more important point, that this inorganic inclusion body, like which is containing polymethaphosphate or volutin or metachromatic granules, they are found in certain bacteria such as your Corynebacterium, diphtheriae. So these are the inclusion bodies, all about inclusion body. Next, that is mesosomes. Now, so what are the mesosomes? So mesosomes are just invagination of your plasma membrane in the form of vesicles, tubules or lamellae. One more important point, that they are more prominent in gram-positive bacteria. Mesosomes are more prominent in gram-positive bacteria. Then coming to the location for the mesosome. So mesosomes are often found next to septa in a dividing bacteria, or sometimes seen attached to the bacterial chromosome. Okay, sometimes it is seen as attached with the bacterial chromosome, and if you see bacteria at the time of dividing, then it is found next to septa. Clear? Now coming to the functions, so I will write here. Okay, it is attached to chromosome or with septa when you are seeing at the time of dividing bacteria. It is the invagination of plasma membrane. Remember this. Now functions of the mesosomes. So the first function, that it is the site for respiration, bacterial respiration. Okay, this mesosome possesses respiratory enzymes, and these enzymes are just analogous to mitochondria of eukaryotes. Clear? The enzymes which are present in mesosomes are analogous to the enzymes which are present in mitochondria. Next important function, they involved in cell wall formation. Okay, they also play a role in chromosomal replication. They also play a role in distribution of chromosome to daughter cell. Okay, so these are the four important functions of the mesosomes. First one is site of respiration, cell wall formation, then chromosome replication and distribution of those chromosome to daughter cells. So this is all about your mesosomes. Now coming to the next, that is your nucleoid. This is the fourth component. Clear? So bacteria do not have a true nucleus, means true nucleus is absent in bacteria, but the genetic material is located in an irregularly shaped region which is known as nucleoid. Clear? There is no nuclear membrane or nucleolus. Nuclear membrane and nucleolus is completely absent in the case of bacteria. Clear? Now some important points. Bacteria possess a single haploid chromosome. Very important. Single haploid chromosome. Bacteria possess a single haploid chromosome comprising of supercoiled circular double-stranded DNA. This is important, circular double-stranded DNA. So nucleoid consists of a single haploid chromosome which comprises of supercoiled circular double-stranded DNA. Okay, and one important point, this DNA lacks basic protein. Very important, this DNA lacks basic proteins, because basic proteins, as you have read, that eukaryotic organism nucleus contains those basic proteins, but in bacteria these basic proteins are absent. Clear? The next important point, but one exception I need to mention here, some bacteria have a linear chromosome instead of circular. Linear chromosome, and some have two chromosomes instead. Okay, we have discussed single haploid and circular, but some have a linear chromosome and some have two chromosomes. Example is your Vibrio cholerae. Okay. Now bacterial DNA divides by simple binary fission, and the nucleoid can be seen by electron microscopy, okay, or on staining with a stain, such as, this is very important from MCQ, that is Feulgen stain. This stain, that is Feulgen stain, is used for staining nucleoid. Okay, stain is your Feulgen stain, very important, Feulgen stain. The next, bacteria also possess extra chromosomal DNA, which is known as plasmid. So the two important, very important points, that it lacks basic protein, but Vibrio chloride is an exception in regard to that, and the staining is done by Feulgen stain. Clear? Now we will look for, so this is about your intracytoplasmic, this is cytoplasmic matrix.